Hi, everyone. Most of you already know me, but in case you don't, I am Dr. Christina Cardona from the Essex campus teaching environmental science and oceanography. I have my friend here, Dr. Jan Vicente, from coming to us from Honolulu. Well, no,
Yeah, Honolulu.
Honolulu. Yeah, I thought you were like Coconut Island or whatever in Hawaii. Uh, so it is bright and early, 8:30 in the morning for him, but he's here, ready to present on marine sponges and his work there. And I asked him to come here in association with the Community Book Connections, Wild Tongues Can't Be Tamed. But there's not much introduction here for most students here are in my environmental science or oceanography classes. So I will let you go ahead.
Awesome. Well, yeah, thank you all for being here and Christina for offering this opportunity. I basically live for these moments where I can talk about the work that we do, and I love to teach and mentor students as much as possible. So every time I give a talk, I I try to leave students with um, some motivation to come and do, if they want to get some research experience with our work, I try and get them to work with us if possible in any kind of way. Unfortunately, because of the distance, it's harder with Baltimore, but who knows? Maybe we could figure out something. I don't know. So, yes, my name is John. I did my PhD in the Institute of Marine Environmental Technology, actually, in Baltimore, and Baltimore has a special place in my heart. Lived there for 6 years. I actually lived with Christina, in her basement for like a couple of those years. So, thanks to her, I was able to um, travel to places in the Caribbean that I wouldn't be able to afford otherwise. So thank you, Christina, for helping this research today.
Tell
Um,
them how bad our basement is.
it was a great basement. I loved it. It was very cozy. So today I'm going to talk to you about sponges, and I've been working with sponges since I was 21, I want to say, and I'm 41 now, so it's been 20 years. I'm originally from Puerto Rico. And a lot of the work I did in Puerto Rico was natural product chemistry. I moved to Wilmington, University of North Carolina, Wilmington, to do my master's with bacteria that live in the sponges that we can culture. And I fermented a bunch of bacteria from sponges. And we were isolating all these antibiotic compounds. And I was very intrigued by the amount of bacteria that lived in sponges. So, with that, I got very motivated to do a PhD, and I went up to Maryland in the lab of Professor Russell Hill, who was a guru at culturing bacteria and understanding the relationship between bacteria and sponges. And then I applied for some scholarships as a postdoc, which got me to Hawaii. And when I moved to Hawaii, I discovered something completely different than what I was used to. And I hope to teach you a little bit about that today. Um, so before I begin, it's very important to understand that sponges are at the very base of the Metazoan tree of life. Some fossil records are said to be more than 850 million years old. So they've been around for quite a long time and haven't become extinct for more than six extinction events, right? Apparently, more simple, mobera, like Hawaiians say here. Because you're able to, apparently, sponges have figured out a way to survive many of these extinction events. And they have a very basic body plan. Um, and sponges, with their basic body plan, have been are remarkable sources of nutrients in coral reef habitats. They have an incredible ability to pump and filter water, as you can see here in this video. If you look at a sponge, without this fluorescein dye, you wouldn't be able to tell how much it's pumping. But this is an artificial dye that we put around the walls of the sponge, and in a matter of 30 seconds, This dye gets taken up through the walls of the sponge and gets pumped out. And with that, they retain like 99% of particles, bacteria, zooplankton that's in that water. And the exudates are basically almost sterile. Um, they are feeding on several things here. They're feeding on bacteria. They're also feeding on dissolved organic matter, which is their number one food source. Um, and this water comes in contact when the water is coming through the sponge wall, um, coming in contact with a lot of bacteria that live inside the sponge host, right? So a single sponge species can have up to 14,000 species of bacteria inside the tissue. It's unbelievable. Like the diversity of microbes inside sponges is unbelievable. However, in other species, you can have only a few species of bacteria. So they've evolved different strategies to cope with their physiologies through the evolution of the tree of life, or of the evolution of sponges. So with their relationships with microbes, so this is a Caribbean reef. If you were to go snorkeling in the Caribbean, you would find these massive sponges that are emergent or growing from the surface of the reef towards the sunlight. And if we were to look at some sections with a microscope of the sponge tissue, we would find sponges that have adapted relationships with lots of bacteria, like these high microbial abundant sponges, or relationships with a small group of bacteria, and those are termed low microbial abundant sponges, right? And so if you look at these micrographs, so these are transmission electron micrographs that are taken with a electron microscopy. You can see here that these big circular globular cells are sponge cells that are in a environment devoid of bacteria. There's no bacterial cells here, basically, and this is almost exclusively sponge cells. However, if we look at this high microbial abundant micrograph of a giant barrel sponge, we'll see that there are some sponge cells, but most of it is bacterial cells that are in the tissue. So with that comes a slew of metabolic processes that the sponges can do. Okay, so if, for example, if these sponges that harbor a lot of cyanobacteria, for example, they can fix nitrogen, they can fix carbon, they can process ammonia. Um, while the sponges that don't have any bacteria rely more on heterotrophic feeding, which means that any particles in the water column get captured by the quantocyte cells in the sponge body and they get digested for food. So these are two different physiological evolutionary pathways that they've developed over time. And through this process, you could see it as a way to offset competition, right? So if you're two sponges that have the same physiological demands and you're in an environment where those food sources are limited and you're competing for that same food source, there's going to be competition. Well, this is a way or a strategy to offset those pressures, right? If I associate with a different set of microbes and this other sponge doesn't associate with microbes, then we will process food completely different and we will target food differently. Okay, so that's basically a little summary of how microbes and symbiosis in sponges is important, right? Now, sponges also play some really important roles in the biodiversity of reefs, right? So when Darwin went out on his expeditions, he could not come to terms with the fact that coral reefs were so biodiverse, but lived basically in a marine desert, right? So imagine these archipelagos, these island archipelagos, just like Hawaii or remote archipelagos. The Caribbean, you can't really think of as a remote archipelago because it's adjacent to massive continental shelves that are pumping in loads of nutrients. Right. So these, when I say remote, I'm talking about thousands and thousands of miles offshore. So Hawaii is a really good example of that. So he couldn't come to terms with the fact that Coral reefs were so biodiverse, but we're living in such oligotrophic conditions. So how is it that biodiversity is sustained in areas that have so little nutrients, right? Well, recently there was a, well, and recently it's more, it's been 12 years now, but 12 years ago, scientists found out that sponges might have an answer to that paradox that Darwin was dealing with. And that's because they found that sponges that live inside the nooks and crannies of the reef, such as this species, Halisarca cerulea. This species is a sponge that's probably the size of my hand, and it is a thinly encrusting sponge. It doesn't grow very big. So very different from the giant barrel sponge that you saw in the other picture, right? This is a tiny little sponge. And over time, scientists are like, well, why does this sponge not grow the size of a car? The reason why that is because if you look at the tissue of the sponge and you look at these quanocyte cells, so I don't know if you can see, but there are these kind of spaces here, right? And these spaces are surrounded by these tiny little cells that have flagella, and that is called a quanocyte chamber because the cells are quanocytes. And quanocytes have these flagella that are um, just like uh, propelling the water into the sponge tissue. And that's their main function, right? What scientists didn't know was that other than pumping water and having that as the sole function, these cells were basically being shed into the environment. So if you put this sponge in aquaria and you monitor the water over time, you'll see that the cells just fall off the sponge. And they do that rather quickly. Think about it in terms of how humans are constantly shedding their skin cells, or our intestinal cells. So every time we go to the bathroom, we're shedding a lot of the intestinal lining of our stomachs and our intestines, right? So all these cells are being shed. Okay, so why is that important? Well, they also monitored that these sponges are taking up this form of carbon that can be metabolized by other organisms other than bacteria. So bacteria, so for those of you who are biological oceanographers, you'll know that dissolved organic matter in the food webs is taken up by bacteria, and the bacteria basically die and they feed the food chain by basically serving as their dead cells are basically food particles for tiny little detritivores, right? So through these mechanisms, we're fueling the food chain. So these tiny little organisms down here are actually feeding on this particulate form of carbon that came from these sponges, or in a pelagic system, comes from the bacteria in the pelagic food system. Now, what's interesting is that sponges do this 30 times faster than the bacteria in pelagic food web system. So this is happening continuously. Like it's unbelievable. This is potentially a reason why Darwin was able, this is a potential solution for Darwin's paradox, right? So you're taking a form of dissolved organic matter that's released by corals, and it's plentiful because you're only competing with bacteria, you're competing with other sponges, and because other organisms can't take it up, they basically recycle that DOM. They pump it out as dead sponge tissue for detritivores to basically feed on and stimulate and fuel diversity with that mechanism. All right, so like I mentioned, those are basically some very important insights of why sponges are so important. And it's basically those small encrusting sponges that do that. So when we look at the Caribbean Reef and where I'm from, sponges look like this. They're massive. They're also contributing to the habitat complexity of the reef. So fishies and turtles and lobsters can hide it actually inside these barrels on the right. Now, when I moved to Hawaii, this is what the reef looked like. So I was like, is there even going to be a job for me in Hawaii? Because the reef is dominated by corals. Good luck if you find a sponge that's emergent and a giant barrel sponge. Now, when I came here, the only reputation that sponges had for the state of Hawaii were invasive sponges, right? And invasive sponges can wreak havoc on ecosystems. So in remote places, you have these encrusting sponges that aggressively overgrow coral. This is the case of a sponge that associates with photosynthesizing cyanobacteria. It can grow very quickly and kill coral. The same happens with this boring sponge. So sponges can actually dissolve the calcium carbonated tissue of corals and basically overtake the habitat. And in Hawaii, you have this case of this sponge that's red that can grow inside the nooks and crannies of these finger coral or Parietes compressa that we call it. So when I moved to Hawaii, they're like, well, if you want to study sponges, you're going to have to study these invasive sponges because there's really not much else that you can study. And I was like, well, I don't know about that. We'll see. Um, so when I first came into Hawaii, I was interested in studying how these invasive species were growing on the reefs, and we found that tiger cowries were actually a really good control system for eating these sponges, right? So here's some tiger cowries eating on an invasive sponge. You can see that they're pooping the sponge, and the detritivores are actually vacuuming all that detritus. So this is kind of a different view of the sponge loop. Instead of the bacteria kind of processing and shedding the cells, these are the cowries doing that functional role and pooping sponges for detritivores to eat. But basically, yeah, that was my first project here in Hawaii. We're looking at how tiger cowries, which is the biggest shell of any tiger cowries in the world. It's the Hawaiian tiger cowrie. It's unbelievable. They grow the size of I want to say a softball. And so we're encouraging the state of Hawaii to, there are no state laws that protect them right now. And so we're trying to see how they can be protected under state law so that they don't get uh, purchased by all these jewelry companies to make a spoon out of them and sell them on Instagram or whatever. Turtles also feed on sponges. So also discovered that they're actually an important food source for green sea turtles. So here you'll see all these pigments coming out of the mouth of a sea turtle. And algae doesn't produce those pigments, it's the sponges. So these tiny little sponges that look like algae that grow in the sand that are boring sponges are actually contributing to their diet. Okay, so I mentioned how we couldn't find these giant barrel sponges. And I was like, well, what about the sponges inside the caves, right? Sponges that are inside the cryptofauna. And oh my goodness, we found so many species, right? We might not see sponges on top of the reef, but they are everywhere inside of the reef. And if you remember from that slide that I showed that has a lot of detritus being produced from the shedding of the cells, live inside cryptic communities. Just like that one, we have hundreds of species living inside these lava tubes in Hawaii. We also find these sponges that are used natively by indigenous Hawaiians to sand canoes. So these sponges are called stony sponges. And they're also producers of anti-cancer compounds. Um, and so these sponges are usually found very deep in the Atlantic. So you can see here that in 2,400 feet, scientists have found on the right similar sponges to the ones we find here. But here we find it only in 25 to 30 feet. So we're trying to protect those as well from over harvesting for cancer research and all of this kind of incentives. But we do find these habitat forming stony sponges inside these lava tubes as well. And we have students that have dedicated their thesis or master's thesis to describing them. She has actually found that these species are closely related to other species in French Polynesia, also found in lava tubes. And these are museum collections from hundreds of years old. So I work with the Bishop Museum as a curator of sponges. And these are basically records that are hundreds of years old. And these sponges have been found recently and shows that they're still around. They haven't become extinct. But for one species, we found actually that that species might have gone extinct. And that's why museum collections are so important, right? So collections from decades and decades ago, if you find them back then and you don't find them now, it's an indication that they're potentially being harmed by climate change or some changes in the environment. So in Hawaii here, we have a lot of issues with groundwater discharge from land development, and water that's not treated that goes into the ocean and just basically creates diseases for organisms, and sponges are very sensitive to these. All right, so because I saw this diversity of the sponge cryptofauna, I was very intrigued to understand what they were and what they were doing. Um, and so the problem with the cryptofauna is that the sponges are hidden, right? So in order to get to them, you either need to blow up the reef, which we're definitely not going to do, or basically dive inside the nooks and crannies, which we can't do because we can't fit inside the little fingers of the corals, right? So I was interested in documenting whether or not this diversity of sponges was taking up dissolved organic matter and feeding all these tiny little critters, right? But in order to do that, we need to develop a technology that allows us to access that hidden habitat. And it's important to understand what the cryptofauna is, right? So if you look at the reef framework, like I said, you can't really fit your fingers inside the nooks and crannies of the reef. And there's all these niches of what cryptic habitat is. So these are all little letters on a coral reef that indicate what cryptic habitat means, right? So on A, you have rubble, loose rubble. On B, you have like shaded environments that are caused by massive coral. On E, you have the nooks and crannies. F, you have the nooks and crannies of the reef. And other examples of what the cryptic fauna is. So it's really delicate system, right, to get in there. And how do you study it? Well, for years, scientists have developed this technique to study what the diversity of organisms can be in a cubic foot of coral reef. And this is important, right? Because biodiversity is the currency, species diversity is the currency of conservation. If you want to conserve an area, you want to make sure you're conserving the right habitat that has the richest biodiversity, because biodiversity is indicative of a very healthy, functioning habitat. So, if you were to go snorkeling in coral reefs and you were to observe the fauna of coral reefs, you would find that only less than 1% of that reef fauna is detectable. Okay. You would only see fish, and you would, if you do a survey of that habitat, you would only see fish, coral, things that are emerging from the reef. What you wouldn't see is that 99% of the diversity is found hidden within the cryptic benthos. Okay. So these are just examples of how much of that diversity is hidden within the cryptic benthos. All right. In order to detect that diversity, we use these autonomous reef monitoring structures, right? And it is a very sustainable way of accessing the reef habitat diversity. We put these arms units out on the reef, and think of it as an apartment complex of coral reef organisms. They're basically a stack of settlement plates, and in between settlement plates, there's areas that have flow for water to go from one point to the other. Some are semi-closed to create different niches. And over a span of 2 years, when you leave out the arms on the reef environment, this is what it looks like. It's been embedded with all these reef critters. We put a milk crate with a thin 100 micron meter mesh bag around it to keep all the motile critters like fish and crabs retained within these boxes. You bring it up to the surface, and all of these floors of the apartment complex get disassembled, right? And what we do is that we pick out all the motile stuff and we separate all these motile creatures from all of the sessile stuff that's growing on these arms plates. So the way we separate the motile creatures is by using three different filter types, or these are sieves. So we're sieving out all the organisms based by size class. We have the very large organisms that um, you can just pick out from the tray, process the water through a smaller size sieve, 2 millimeters of size, and you can see this fraction. And then we do a 1 millimeter, sorry, a 500 micrometer mesh to get this fine sediment to get little worms and other cryptic critters. Now, the way that scientists have been processing the diversity on the plates has been gut-wrenching for me because what they do is that they scrape it with a paint scraper, they put it in a blender, and they fix it in ethanol. And you can see from this picture that there's at least 10 sponge species on this plate. And what they do is that they metabarcode everything that's on that plate. The reason why it's gut-wrenching for me is because I know that probably every single species on that plate is a new species because we do not have a database for these critters that are living inside the reef environment. So when you do this sequencing technology, what you're missing out on is the actual barcodes of each of those individual species to actually know what's on there. So let's say you scrape this, you extract DNA from it, you metabarcode it. Um, there's going to be a lot of data in your sample that's going to be eliminated because it doesn't match anything that's on your plate, right? So that's where I come in, and I basically identify all the species that are in there and provide those species with barcodes and make sure that the database can be usable for bioinformaticians to detect cryptic reef diversity. So we basically have used these standardized structures to monitor the diversity of the reef. We know they harbor great diversity. They're great at recruiting the sponge fauna. And we can answer some really cool questions about what is the level of endemicity of species on the reef environment? Do we have sponges that are introduced? Do we have sponges that are only found within cryptic environments of that specific place? Um, if we monitor that apartment complex over time, can we see different temporal patterns of richness? What is the amount of carbon that is being turned over by this community over time, right? And we're doing this all without destroying the coral reef environment. So it's great, right? Perfect tool to use. So the first question I wanted to answer was, well, how is the diversity in these units different from the diversity that we find in all of these 35 sites in Hawaii? So this is the Bay of Kaneohe Bay, and this is the island, Coconut Island, where I work every day. We basically have an island that is located on top of a patch reef. So we're literally working on top of a coral reef ecosystem every day. It's pretty awesome. And by the time I came in at a time where scientists were already conducting experiments to look at how climate change was affecting the biodiversity of coral reefs using these arms, right? So here you have arms in the different colors, you have different treatments, and the different squares are different replicate tanks where we put arms inside of them to look at the change of the community in the presence of heat, acidification, and the combination of heat and acidification compared to the control, okay? And these are arms inside mesocosm tanks. We also had arms on the reef to look at how the mesocosm condition was affecting the oral recruitment of the arms in relation to the natural reef environment. Okay, so the first question is to look at how the diversity in the arms is different from the diversity in the reef. We surveyed 35 sites. When we looked at the reef environment, we sampled over 150 samples that we barcoded with CO1 and 28S gene markers. And for sponges in the arms, we collected way more sponges because we had so many more specimens to work with, which indicates that we actually can access the cryptic reef community. So the sponges on the reef were collected by hand, by flipping rubble, looking underneath the overhangs. Really, we're not really getting into the deep matrix of the reef. But with arms, because the arms is actually simulating a reef environment where you can actually dig into the deep environment, we were able to access way more sponges. So this is what it looked like to process the arms units over time in these mesocosm tanks. These are the tanks on the left. We disassembled the arms. Here's me photographing and subsampling all the sponges. And we also took a overhead image so that we can identify all the species over time. So we deployed, we image analysis, and we tracked the growth of all these different sponge species in the different colors over time. Okay, so it's very tedious, as you can see, like these are centimeters and even millimeters of size of sponges. These are tiny little critters that are growing on these arms plates. Okay, so the first thing was to create a database for these sponges. So we collected the sponges, we stored samples for DNA, we fixed samples for histology and for vouchering in museums. We identified the sponges either through histology or through the skeleton morphology of the species that were growing on there. And then we do a scanning electron micrography for the spicules that are making the skeleton of the sponge. The same organism is anchored with DNA barcodes that are then compared to global databases to look at whether or not these species are from Hawaii or from a different place. And so with that, we're able to compare this diversity with the rest of the world. So that's how much work goes into the identification of a species, right? So then we can identify the species and trace the percent cover of those species on the actual arms plates to look at how these species are growing on the arms plates over time and over the different treatments. So this is a phylogenetic tree on the left showing that every single species that you see here is unique. In the middle panel, you can see how these species are distributed on the plate, and we can calculate percent cover and just a morphological diagnosis of these species to document their diversity. And with that, we've actually had students that have become pros at histological slides. And here you can see how there's all these histological sections of species and how the skeleton actually gives us a lot of characters for the species that we're working with. And we're able to compare the data that is extracted from metabarcoding analysis, and how the contribution of us single-handedly subsampling every species compares to the yield with metabarcoding using metabarcoding primers. So now we have a way to show how much is being missed by this metabarcoding technology when we go in there and we subsample sponge species. And what we found was that when you do metabarcoding at the class level, detection is completely lost, right? So here for a class of calcareous sponges, not a single species is detected with that metabarcoding primer. However, our single-handedly collection of sponges detects that there's actually these many species, but when we do metabarcoding, all of this diversity is lost. So by looking at this from the lens of integrative taxonomy versus just relying on metabarcoding data, we've discovered that there's a lot of holes that need to be filled with the knowledge of taxonomists. And we have a student who's graduated and defended her thesis where she's developed a primer that actually is specific for sponges and works better for sponges than these traditional primers that we've been using. And this new metabarcoding primer is probably going to revolutionize the field of eDNA metabarcoding technology for reef critters, because we not only found that it works for sponges, but it works for a bunch of other phyla that the traditional metabarcoding primers did not pick up. Sorry, excuse me, I'm going to sneeze. Excuse me. Okay, so with the development of that primer, we've actually gone on to expand our research a little more, and we're looking at the diversity of arms, not only in Hawaii, but in the Marianas, the Coral Triangle, the Line Islands, and American Samoa. So that is a project that's ongoing right now. Okay, so back to that original question where we surveyed 35 sites and we wanted to compare the diversity of the reef and the arms, right? What did we find? Okay, so we found that in arms, so here you have these little diagrams here, um, this Venn diagram that shows that the reef had a unique microbial community. The arms had a unique microbial sponge community, not microbial community, sponge community, and that only 38 species, we're only recovered 38 species shared between both efforts. Okay. So we're definitely collecting something different with the arms and something different with the reef that we can't access if it wasn't for the arms. Okay. And what was really remarkable, though, was that the diversity that we found in only 50 meters square of arms material of these PVC pipes accounted for greater than 50% of the diversity found in more than 1,700 meters squared of surveyed reef environment. Okay. So the arms are only deployed at this site. We surveyed this entire bay. Okay. And the sponges accounted for more than 50% of the diversity was accounted for by the arms. It was a remarkable, astonishing result when we saw that. So it doesn't matter how well you survey a reef. If you don't have these arms, basically, you're not going to really get the diversity that's in the cryptofauna. Another remarkable thing that we saw is that different from the reef environment in the Caribbean, where everything is found on top of the reef, right? All these emergent sponges are found on top of the reef. Only 20% are surface sponges, and 80% of the diversity is found hidden and embedded within the reef environment. You cannot find these sponges if you don't look under the rocks. So basically, through the history of Kaneohe Bay, we discovered that we added more than 150 new records with this work, and that more than 100 are new species. So we think we're surveying the reef basically by looking at the surface community. We weren't. We are missing an astounding diversity that lives there and has been completely ignored over all these years. And so this is remarkable, right? Because when you look at the diversity of porifera in Hawaii compared to the world, this is what we knew in 2013, that there are only 61 species. Okay. We have found that in one bay, in one island, the diversity is more than 180. Okay. So this map is definitely going to change in the future. And the reason why we have so many diversity in the North Atlantic is because of sampling effort. There's been a lot of expedition throughout the years in the Northeast Atlantic and the Caribbean, but these remote places in Oceania are completely understudied. Okay. So we have a lot of work to do. And the problem with Hawaii is that we get a lot of our species, some of our species from the Caribbean, from the Mediterranean, from the Indo-Pacific, and Central Indo-Pacific and the West Indo-Pacific, right? And that's because we have a lot of shipping traffic, and we have a lot of debris that is being swept into our monument and our islands from Japan and from Asia. We're also losing taxonomists, which is a very, very hard and gut-wrenching thing to experience. We have all this diversity of species in Kaneohe Bay. These are just examples of some of the beautiful sponges from just one order. There's 19 orders. Just one order has all these species. And luckily, we have the next generation that I'm training here in Hawaii that's allowing for this diversity to basically get expanded. And hopefully this creates more awareness for people to put in more work. What about, okay, so now we know that there's a huge diversity that's picked up by the arms. What are their functional roles, right? So we know there's diversity. What are they doing for the reef environment? All right. So first we look at temporal fluctuations. So for this project, we used the same setup for this experiment, right? And we compared the arms that were on the reef to the arms that were in the mesocosm community with ambient conditions, okay? And we were just comparing richness, diversity, and abundance over time, over 2 years. All right, we found lots of diversity within these very dominant classes of sponges, right? There's three dominant classes, the demospongi, the hemoscleromorph, and the calcarea. The calcarea only produce calcareous skeleton, right? Like corals. Homoscleromorpha and demosponges can produce skeleton out of silica, but they can also lack skeletal structures and have just a structure, a skeleton structure made out of fibers. And what we saw was pretty interesting. So this is a snapshot of arms plates in the mesocosm community with an incredibly rich community of sponges. And we traced that community of sponges over time from June 7th to November 20th. And what we found was that the sponges reach a peak diversity after a year, but then that diversity starts dwindling. And we were wondering why is that? Why do we have such a quick growth of diversity and then that diversity just dies naturally? Well, you can think about it kind of as in ecological studies, classical ecological studies in terrestrial systems, we see how weeds and pioneering species create this incredible diversity because they are selected species, are really quick at dispersing and growing quickly. Then you have the arrival of dominant species like big trees that dominate and create shade over the understory. These weeds die, and what's left is this climax community of these K-selected species, which are slow growing but dominant, right? Species. Now, in the early studies of succession and the application of this in the terrestrial in the marine environments, scientists said, well, the trees or the dominant species are those species that are encrusting, and the weeds are those species that are solitary. And what I mean by solitary and colonial is basically solitary are species that are erect and single, and colonial kind of spread. Okay, so the colonial species are the white arrows. You can see how they're just spreading and overtaking the environment. But these solitary species don't really take a lot of real estate and are just like outcropping on the reef environment. So we're basically seeing the same thing, but in the marine environment with sponges. And what we did was that we counted, so we compared the diversity in the mesocosm with the reef environment, and this is what we found. We basically found way more diversity. So here you have species richness in the y-axis and the number of individuals on the x-axis. And we see that in the mesocosm, way more diversity than the reef. All right. And the reason for that was because the reef wasn't recruiting some sponges in Calcarea, some sponges in Suberitida, and some sponges in Tetractinellida. And all of those three are solitary sponges, or sponges that don't spread. So what we're seeing is that with time here, so number of species on the y-axis, the date is on the x-axis. When we compare the mesocosm in blue, and we compare the reef in orange, we see that in the mesocosm, we have a climax diversity, and then the diversity drops. The diversity in the reef environment hasn't reached climax yet after 2 years. Okay. So what we think is happening is that by removing the arms from a natural reef environment and putting it in a mesocosm, it's actually experiencing a pioneering stage of succession that the reef community would not be able to experience because it's already at climax. If you put an arms on the reef community has already experienced a climax community, and what you're going to get is recruitment of all these dominant species. But in the mesocosm, away from a climax community, you actually see that there's a climax of the community, and that climax community dies. Okay. And now it's kind of more acclimating to a to the reef community, reef climax community over time. So that's what we think is happening. It was really interesting to see this dynamic and to see what the heck is happening in a mesocosm that's away from the reef environment. Like, wouldn't we see more diversity on the reef natural environment? It was completely the opposite, but we believe that this is why we saw what we saw. There were different sponges responsible for the shifts in that diversity and abundance. And it was basically those calcareous sponges and those and some sponges that are demo sponges that belong to the order Haplosclera that were causing those shifts in the community. Some others had a similar pattern as well. What's interesting about the calcareous sponges is that they're all solitary. You can see them. They're all little tiny sponges that don't take a lot of real estate. And we also found that the Haplosclera have a lot of reproductive output. So that's very uh, typical of a weedy species, right? So the weeds are always dispersing their seeds. These sponges are always dispersing their eggs. Okay. Now, what's interesting is that um, we were also, because this was the arms were inside these mesocosms, and we were looking at the impact of climate change. We also were able to monitor that impact of climate change. And what we saw was that the sponges community is pretty resilient. Only one group gets blasted by the two factors, and that is the Homoscleromorpha. Okay. There was basically no species detected in the acidify treatment, and that the overall diversity decreased in relation to the control. What was even more remarkable is that the calcarea, which are calcifying organisms, had higher diversity in the acidification treatment than all the other treatments. So these sponges are resilient. They're going to endure climate change. And um, uh, they're basically going to continue contributing um, to the overall turnover of those nutrients. So when these sponges die, to reach that climax community and to succeed after that pioneering species, all those nutrients are going into this system inside the reef environment. So how does that tie into the sponge loop and what we know about the nutrient dynamics, right? That I mentioned before, right? How are these nutrients basically contributing to the sponge loop? Well, if we look at the sponge loop again, uh, we talk about DOM being taken up by sponges that get turned over and fixed by these quantocyte cells, which are contributed to the particulate food web that fuels higher diversity. Well, what we've added to this whole thing is that potentially um, these sponges that are pioneering species are also contributing to the sponge loop by just dying over time and contributing the dead sponge cells that are actually preyed upon by these detritivores as well. So not only can the sponge loop be seen by the recycling of cells, but also by the short-lived life cycles of these weedy species. And so we've had scientists actually come to Hawaii and determine whether this DOM uptake is actually happening in Hawaii. And the answer to that is that it is happening, and that detritus is being produced by these sponges. You can see the detritus amount of sponges being way significantly more than the control here in these experiments. We basically put these sponge toilets under the sponges in arms over time to look at how much of this detritus was being produced by the sponges in comparison to the seawater. And with that, I thank so many people that have brought me here, from taxonomy courses, sponge conferences, collaborators. Um, um, there have been several research opportunities for undergraduates in Hawaiian from Native Hawaiian communities that have partaken in this work. And if it wasn't for them, a lot of this work wouldn't have been done. Um, and with that, I'll take any questions. Yeah.
If anyone has any questions, you can raise your hand or type it in the chat. I also have a survey for you to fill out. Students that enter or submit the survey are entered to win $25 gift card. We need survey responses to keep programming like this going on at CCBC.
So I have a question, but it's too much for me to type. Can I just ask it?
Yeah, yeah, go ahead.
Yes.
Okay, great. First of all, thank you so much for this presentation, bro. It was very informative. First and foremost, as all of these sessions have been. My question is always, I'm kind of like a conspiracy theorist, I think, and I always have thoughts when it comes to hearing about things in the ocean that um, um, are just being discovered. Um, how closely, and you know, since you study fossils, how closely are the things that are being discovered related to something that's, you know, I think we have like normal animals or animals that have become more normalized, but are there any abnormalities or any different characteristics that you're seeing in the new species
Yeah, that's
that
a good
are just
question.
like normal? Yeah.
Yeah, that's a good question. Yeah, so that's a good question. We with this diversity that we're uncovering, there are some really weird sponges that it's part of a different project. We basically targeted the community of these lava tubes. So in Hawaii, you have a lot of everything's volcanic islands. And when volcanoes erupt, you have these lava tube formations that allow a cryptic environment or cave environment so that uh, the cryptic community basically develops in these cave environments. And we found a sponge that looks, I thought I was collecting a sample of sand. And I was like, this is weird. This is definitely a sponge. This is definitely a sponge. But the amount of sand that it had accumulated was something I've never seen before. So for many years, um, I was trying to figure out what it was, and luckily, we looked at the DNA and we knew it was a sponge. And I looked at the skeletal structures more closely under the microscope, and we actually found the characters for sponges in those sections. And it's just a sponge that loves to accumulate sand kind of to develop its skeleton. And it was really cool to see that. And it's been one of the most kind of extraordinary things I've seen in terms of our collection. But other than that, nothing more out of the ordinary.
So is it safe to say, and again, I'm just asking from the conspiracy theorist standpoint, all of, and please don't judge me too harshly, but all of my favorite movies of you know, if it's a doomsday movie or it's even something Marvel based, always speaks about life in the ocean that's undiscovered or that's not it's not normalized. It's like the attributes and things that are discovered are normally not put out into the mainstream of like this is what we're just even what you just said that can even be possible from what I'm knowing from sponges from the limited knowledge that I've got gathered from sponges from you know this class. Of course, I don't know as much as you do. I'm just saying why aren't those abnormalities like um what is the percentage that I saw? We only know 6% about the ocean. You know,
Yeah.
how is that information then passed on to, I guess, everyday people?
Yeah, that's man, I really appreciate your question, and I think it's very important, and it's actually kind of an earth-shaking question because your question is an answer to us as scientists not doing enough to communicate our science with the general public. And right now, when all this money, all these cuts are happening at the government level because scientists are not doing their job and or whatever, I I think a part of it is because I mean, obviously, you know, there's a lot of people to blame, right? But we take responsibility. I think we should all take responsibility for that as well because we get lost in the sauce of science so much that we forget that we need to communicate our findings to the general public. Um, at the same, we need to dedicate the same effort that we're dedicating to the discovery of species. We need to dedicate to getting the information out. Because if not, you're going to have situations like this where people are like, well, I don't really believe it because I don't see it, or I'm not seeing any studies coming out of that. And unfortunately, journals and papers that are written for a targeted audience and not really necessarily for the general audience. So I really appreciate your question, and I think that the answer to it is that yes, there's an incredible diversity that we have yet to to understand, quite honestly. And and I know you're just trying to trust my word of mouth right now, but as many decades of experience studying this, I can tell you that we know so little of even the group of sponges, something we've been studying for 100 more than 100 years. We've thought that we figured out how much diversity is in the ocean, and this little snippet, I hope to convince you that this little snippet of data is just a little snippet of data, right, from one island, from one reef in the island of Hawaii, in the island of Oahu, in the island of Coconut Island in HIMB, where I do my research. That is just one location out of a vast ocean of thousands of islands, right? So we've increased the diversity by fourfold only in that reef environment. And that's just me, a guy who's decided to look at the sponges in Hawaii. Like anyone could do this, right? So um, it is remarkable the diversity that is out there, and I hope that I wish we do better, man. I wish we do better in the future so that people like you get convinced that yeah, this is something that's worth pursuing from a research perspective. I hope I answer your question. I don't know if that helps.
Absolutely. And thank you for being so transparent.
No, I love it.
See a
Yeah.
question on the chat as well. I'm curious your thoughts on
Yeah.
harvesting sea sponges from loofahs or body products. Is there a way to harvest these sustainably, or is it inherently harmful for the species?
Yeah, that's a really good question. So interestingly, the Greek community for thousands of years in the Mediterranean and in Florida, actually, if you go to uh, Tarpon Springs in Florida, you'll see that there's a big community and that people are growing and farming sponges for the purpose of distributing them on Amazon and all that stuff. Obviously, overharvesting is an issue, but I think the farming industry, the aquaculture farming industry of these sponges is pretty good. The only issue is that some of these sponges are very sensitive to warming, to ocean warming. And actually, my dad, who was a sponge taxonomist and sponge biologist in Puerto Rico, documented this mass extinction of species in Puerto Rico. And it was these sponges, these bath sponges. They're very sensitive to warming. When he documented that mass extinction, he gets an email from a guy in the Mediterranean saying, "Oh my goodness, we're noticing the same thing in two different ocean basins. These sponges are actually becoming extinct. They're very sensitive to warming. So, if anything is harming this kind of community, it's climate change. And luckily, sponges are not that popular in the market that they're gonna get over harvested unless obviously we can over harvest anything we want. So it could happen. But yeah, I hope that answers your question.
And there's another one here. How would all the government cuts under this current administration affect your research?
Oh man.
That's opening a can of worms.
Yeah, we let's just put it this way. My wife works for NOAA and every day she checks her email to see if she got fired. Um, the budget cuts are still happening. And yeah, how is it harming it? Well, basically, if right now NSF, who is the prime and sole funding source for this project, Um, their cuts have experienced a 50% reduction, and the amount of scholarships that go out to my students have been severely gutted. In this last call for graduate student fellowships, usually it's hundreds of fellowships that are giving out, and they only awarded nine, for marine biology specifically, nine in the entire nation. Okay. So if we don't have the students, for us to mentor or to teach the next generation, you're basically completely cutting out our ability to document this value in the ocean. Not only that, but the administration wants to move into mining. And the way that mining works is you put a little trawl net that is like a vacuum cleaner of the ocean, and you basically are gutting the seafloor and the seabed environment, from all the diversity that's found. And they don't even care about the diversity. They just want to look for the tiny little metals, right? And so forget about it. Basically, forget about anything I'm talking to you right now, which is really sad, right? Because we just discovered this. And it's like, well, look at the value that there is, right? And people are completely unaware of it. And we need to just advocate more for the work that we do. And we basically need to find new avenues to funding this kind of research. There's billionaires out there. There's a lot of private foundations, and perhaps the federal government right now is not the way to go. So right now we're all hurting, and I'm just one of thousands of scientists in different areas of research that are getting severely, severely, severely hurt by this. And what's very frustrating is that I've dedicated my whole life to this, right? Uh, Scientists at NOAA have dedicated their whole life to this, and they've been stripped from this. You know, they are the experts. They are the ones that have the body of knowledge that no one has. And it is just gut wrenching to know what's happening right now. Yeah, it's really hard, really hard. Yeah.
I do want to point out that a lot of this is temporary, and a lot of this changes with the new administration and
Yeah.
new politicians. So I don't want those of you that are going to the sciences to be disheartened or give up, because I do think like things are bad right now, but they won't stay bad. Things
Yeah.
fluctuate, times change.
You take it day by day. And thank you, Christina, for of us, reminding us of that. You gotta take it one day at a time. And the way that I'm dealing with it is exactly what just take it one day at a time, one day at a time. Put your work out there. Keep on believing that your work is important. Advocate, write letters to your local state representatives, and let's just do our best.
Well, thank you so much, Jan, for being here, and for all the people that came. I know some people already left, but thank you for everyone for being here. And I hope that we'll have Dr. Vicente back here sometime in the future.
It's
Thank
my
you.
pleasure, Christina. And yes, reach out to me whenever you need.